Thank you for the nice introduction, but if you don't mind, I have a brief introduction myself before I start my presentation. I want to say that I'm a molecular biophysicist and biochemist. My expertise is lipids, and I joined as a postdoc Laboratory of Lipid Complexity, as you said, at Weizmann Institute of Science. And this laboratory has been studying lipids for more than 3 decades. At the Laboratory of Lipid Complexity, we're interested in lipids, lipid metabolism, and lipids' many roles in the different stages of life, from origin of life to the mind-boggling complexity existing currently. Here, the antiome is a concept we developed to better define and study metabolic pathways and their origin and evolution. But today I'm going to talk about origin of life, as you could guess. But more precisely, I'm going to talk about a recent review we published concerning lipids in prebiotic chemistry and in origin of life studies. It cannot be overstated the critical roles played by lipids in life as we know it. And because of that, many postulate that lipids also play major roles during the first steps of life development. Some of these steps regard the chemical non-enzymatic generation of diacylglycerophospholipids, the generation of homeostatic reproducing complexes of repertoires of lipids that allow micellar autocatalysis and evolution, and the stereoselective permeability of lipid vesicles to one of the stereoisomers of ribose, important for the formation of RNA. By taking advantage of our background, we now evaluate the most relevant and/or recent studies addressing each one of these topics and conclude that the success and therefore relevance of these models depend entirely on a subset of carefully chosen conditions and on assumptions that may not be fully validated with a consequent impact on our understanding of the origin of life. But let's look at all these in detail. Today, I'm only going to talk about these 2 topics, the generation of glycerophospholipids and the selective permeability of lipid vesicles, and I will finalize by suggesting approaches to enhance the relevance of these studies for origin of life. Regarding generation of glycerophospholipids, I'm going to address this study from Neil Deveraj's group, one of the latest attempts at the non-enzymatic generation of diacylglycerophospholipid, which was published in 2020 in Nature Chemistry. But first, as a mean of comparison, I want to briefly address the biological, thus the enzymatic generation of a glycerophospholipid. Here's an example, the generation of the simple phosphatidic acid from lysophosphatidic acid and oleoyl-CoA by the action of lysophosphatidic acid acyltransferase. And as you can see, this process is not at all a trivial mechanism. It relies on the very specific positioning of very specific amino acids to accomplish the necessary conditions to allow the nucleophilic attack on the thioester substrate for the enzymatic sterification of the lysophospholipid and release of the CoA. This study took inspiration from the enzymatic generation of the glycerophospholipid, and it is used lysophosphatidylcholine, here with the choline group in green and the glycerol group in pink, and the acyl thioester with the leaving group here represented in orange, to demonstrate a high yield, 80 to 90% yield of non-enzymatic synthesis of natural glycerophospholipids, but with only the ester linkage to the glycerol backbone at 37 degrees in an aqueous environment. This is also consistent with the notion that thioesters may have played an important role in the origin of life. This reaction relies solely on electrostatic and hydrophobic interactions. The positive charge of the acyl thioester head group forms favorable interactions with the negative charge of the phosphate group from lyso-PC due to electrostatic attraction. Once the lyso-PC is acylated at the SN2 position of the glycerol backbone, the positively charged amine is released. So, as you can imagine, the need for such specific electrostatic and hydrophobic interactions greatly limit the kind of reactants and conditions that can successfully achieve a product in high yield. In fact, many conditions were tested, but more than half failed to generate a phospholipid in appreciable yields. The conditions tested which failed included changes to the head group of the acyl thioester to negative or neutral charges and changes to the lysophospholipid add group to lysophosphatidyletanolamine, lysophosphatidyl or lysophosphatidyl. Different pHs were also tested, and the reaction only occurred with good yields under high alkaline conditions, with only trace amounts generated at neutral pHs. For slightly lower alkaline pHs, the reaction time had to be significantly increased. Changing the acyl chain length size with changing the acyl chain length to a size which is more prebiotically relevant, namely the C10, which is also less hydrophobic, only generated reasonable yields if the lipids were close to the critical micellar concentration, which was a very high concentration, 5 and 7.5 millimolars. But besides all these, the most problematic, in our opinion, was this one here, the drastic decrease in yield when a third lipid was added to the mixture. This third lipid is STS, sodium uracil sulfate, a commonly used detergent since it partitions to lipid membranes and disrupts their integrity. Additionally, it has a negatively charged head group. Prebiotic Earth, as you can imagine, had no means to distinguish between useful lipids for transacylation and detergent lipids or lipids with non-favorable head groups. So we question if there would be transacylation at all if a more complex and diverse mixture of lipids would have been used. For instance, what would happen in a reaction mixture which contained both reactive lysophospholipids, such as lyso-PC, and unreactive lysophospholipids, such as lyso-PE, PS, or PG. And in the same way, what would the effect be of adding a mixture of more prebiotically relevant, less hydrophobic, short acyl chain fatty acids, which lack the phosphate group required to form the electrostatic interaction with the thioester acyl donor? These are all still open questions. To finish this section, I want to address the assumptions of this study. Here, I'm not going to address the synthesis of the lysophospholipids and the acyl thioester. I'm just going to highlight that much more work needs to be done to offer the synthesis process of these reactants in true prebiotic conditions, but you can find a more detailed explanations of these in the published manuscript. Here, what I want to say is that not only the Sussex of the reaction is limited, but for this limited Sussex to occur, many assumptions had to be made, namely that lysophospholipids and acyl thioesters were available on prebiotic Earth with the correct stereochemistry, at the correct ratios, and appropriate conditions for synthesis. Also, that lysophospholipids and acyl thioesters existed at high concentrations in a pure form, such that the transacylation reaction was not modified or inhibited by environmental conditions or other molecules. Therefore, the limited yields with other lysophospholipids and acyl thioesters, the lack of mechanistic explanation for the reactions which gave no or little yield, the need for high concentrations of poor reactants, and the lack of systematic information on the effect of lipid mixtures and of other environmental conditions appears to somewhat negate the assertion that this approach sheds light on the origin and evolution of cellular membranes. Next, I'm going to address this additional piece of the origin of life puzzle, namely the stereoselective permeability of lipid vesicles. And to this end, I'm going to address this study from Jacques Sosta group. It is from 2005, but it is a key experiment and the basis for much of the later work. In this study, a lipid model system was generated of fatty acids or deacyl phospholipids using only the stereo conformation that is found in modern lipids, namely the R conformation. When the lipid vesicles were in the presence of an hypertonic solution of sugars or sugar alcohols, the kinetics of vesicle permeabilization was measured. As expected by the Mayer-Overton rule and as shown by these values which correspond to the permeability coefficient of each sugar, the more hydrophobic the molecule, the higher the permeability coefficient. Thus, glycerol presented the highest permeability, and with increasing chain length, it was observed a decrease in the permeability coefficient. When we look at the data in more detail, what we learn is that, for instance, from the vesicles of pure fatty acids, or phosphatidylcholine, or of a mixture of phospholipids and cholesterol, as expected with increasing lipid chain length, there is a decrease in the sugar permeability, here represented by this PS. But the permeability coefficient was the lowest in the presence of vesicles generated with the lipid mixture. The focus of the work was D-ribose because of its importance to generate the RNA. And as you can see here, in fact, indeed, D-ribose presents a permeability coefficient 3 to 10 times higher than other aldopentoses, here in different shades of green. This most likely due to differences in the hydrogen bonds that are formed with the lipids, which are more favorable than for the other aldopentoses. However, the permeability coefficient of the sugar alcohol D-L-threatol is higher than that of D-ribose, as seen here in blue. The higher rate of permeability of this sugar alcohol compared to D-ribose indicates a lack of selectivity of uptake, which could result in competition in biochemical reactions, which in turn would likely complicate the formation of RNA. I want to finish this section also with a summary of the assumptions of the model. Lipids emerged on the prebiotic earth with the correct structural chemistry, example chirality, in the correct ratios and in the correct environment. Lipids were pure with no contaminants, existed in high concentrations, and were unreactive with other molecules in their environments. The temperature, pH, and environment were such that the vesicles were all unilamellar and their size distribution was homogeneous, and the ribose only competed with other aldopentoses for nucleic acid formation. Altogether, we can conclude that the large number of assumptions and the limited number of conditions tested does not seem to support the conclusion that the greater membrane permeability of ribose might have been one factor that favored its emergence as the sugar of life. Our criticism is meant to be a constructive criticism, okay? Thus, we want to highlight the limitations of these studies, but we also suggest approaches to increase the relevance to explain the role of lipids in the origin of life. For instance, it is needed an assessment of more probiotically relevant synthesis for the reactants, determine that conditions for reactant synthesis are consistent with conditions required for enzyme-free synthesis of phospholipids, determine whether reaction side products are detrimental to chemical synthesis of the acylation reaction, and it is important to address the effect of the following parameters: non-extruded racemic or scalemic mixtures of lipids, ions, minerals, and other solutes, range of temperature and pH values, for instance, as predicted in prebiotic oceans and hot springs, the effect of minerals that mimic prebiotic earth crust, but above all, it is important to study the effect of complex lipid mixtures, and above all, of mixtures of more than 3 amphiphiles. For instance, by including those found in meteorites, which are usually up to C12 carbons, they can present branched chains and can be decarboxylic acids. And this is true for these studies, but is also true for many other studies addressing origin of life. To conclude, I want to reinforce the idea that our review and my presentation here today are not intended to criticize for the sake of criticism. Instead, what we intend is to stimulate the debate on whether the assumptions behind each model validate or rather undermine some of the conclusions of the studies, because in fact, our analysis highlights the extremely narrow and limited range of conditions by which an experiment is considered to have successfully modeled the role for lipids in an origin of life experiment. And some assumptions and simplifications inherent in such experimental approaches do not permit determination of mechanistic insight into the roles of lipids in the origin of life. So we suggest that more nuanced experimental approaches are required and that a healthy dose of realism about what empirical approaches are credible and which are not would help in determining the path forward. I would like to finish by thanking all my colleagues at Tony Futterman's lab, the Laboratory of Lipid Complexity, whom participate in the discussions of these topics. And I will happily hear your comments and answer your questions now. Thank you for your attention.